In today's lesson, I want to talk about the transmission of a nerve impulse. So when I say the phrase nerve impulse, what I'm talking about here, you guys, is an action potential. Okay, so when we talked about the action potential in the last lesson, I think a lot of people sometimes get the misconception that this is happening in the entire neuron all at the same time. All right, this is actually happening in a very small little slice of the neuron. And what has to happen is this action potential has to move from one end of the neuron to the other. Remember, neurons have a direction, right? The head of the neuron, the front of the neuron, that's the dendrites. Then goes cell body, then axon terminal, right? So an action potential or a nerve impulse has to occur in one little slice, and then it has to move all the way down the neuron to the end. Okay, so it starts at the dendrites, ends at the axon terminal, and it's got to travel the whole way through. So a traveling nerve impulse or a traveling action potential is called nerve transmission. Okay, and that's what we're going to talk about today. Now, before we do that, I just want to run through this graph really quickly to make sure everybody understands what's going on. So in resting potential of our neuron, again, remember last class I said, guys, you need to know which channels are open, which channels are closed, where is sodium, where is potassium, right? Inside or outside the neuron. So at resting, sodium is piled up outside. Okay, potassium is inside. Some kind of disturbance, okay, in the membrane, some sort of stimulus disrupts the membrane and sodium starts to leak in. We get more positive. If we hit this magic number called threshold potential, okay, the voltage regulated sodium channels open at threshold, sodium rushes in, and we now have a positive voltage inside. Okay, now it just the way things work is at plus 45-ish, plus 40, plus 45 again, don't get too hung up on the numbers, okay, just at a very positive voltage inside the cell, that's when the sodium channels close, potassium channels open, potassium rushes out. Now, potassium channels stay open for a little bit longer, so we get lots and lots and lots and lots of potassium rushing out, which gives us this hyperpolarization. Now, right about here, the cell is then resetting itself. So remember, the sodium has to be pumped out and the potassium has to be pumped in. At this point right here, all the channels, all the gated ion channels are closed. The sodium-potassium pumps are working and sodium is being pumped out. Okay, so any sodium that is allowed to leak in is being pumped out. We called this the refractory period. And during the refractory period, we cannot get another action potential, okay, because the pumps are working. So any sodium that would come into the cell is immediately being pumped out. So we don't get another action potential, okay? This is really important for today's lesson, okay? We have to understand that for 2 milliseconds, a neuron cannot generate another action potential because it's in refractory, okay? So nerve impulse transmission is the action potential traveling along the entire neuron. Action potentials start at the dendrites, move through the cell body, move along the axon to the axon terminal. All right, never the other way. So what I want to look at today is how this occurs. How does this action potential travel? Okay, we understand how an action potential happens in a small slice of the neuron, but how does it actually travel? So remember, when an action potential is occurring, we've hit threshold, the sodium channels open, and sodium rushes in. All right, now remember you guys, this is a cell. A neuron might not look like a cell that you might have studied in Science 10, but it's a cell. It has a cytoplasm. The cytoplasm is mainly water, okay? And as you guys have learned, all through science in elementary school and junior high and Science 10, etc., when you put any substance into water, for example, it will diffuse throughout the water, right? It'll go from high concentration to low concentration. That's diffusion and osmosis. So when sodium rushes into the cytoplasm, it's not just held in place, it's not frozen there, right? What's going to happen is that sodium is going to start to diffuse in both directions within the cytoplasm. Okay? So the sodium rushes in and it starts to diffuse. Now, if we were to look at this neuron here, okay, this little part of the axon, if we were to enlarge it, this is where the action potential is occurring. So right here, we're at about that stage. This is depolarization, right? Depolarization is when the sodium rushes in and the inside voltage spikes. Now, as that sodium rushes in, it starts to travel down the axon. So this right here, what's the voltage of this part of the resting cell? It's at -70. It's resting. Now, as sodium starts to diffuse into this section of the cell, let's look at the cell in little sections, like slices. Okay? So this slice right here is depolarizing. This slice right here is resting. But as the sodium starts to diffuse forward like this, what's happening to this -70 number? It's getting more positive. -69, -68, until it hits what? Threshold. And then what happens at threshold? Sodium rushes in, and the second slice is now going to depolarize. Now, remember, the graph we just looked at, what comes after depolarization? Repolarization, and then hyperpolarization, okay? And that's known as refractory. So this section of the cell right here, you can see potassium is rushing out, right? Sodium is in here, and we have refractory. So this is in refractory. Okay, so for that split second, this part of the cell cannot have another action potential, right? So we're looking at it in slices here, right? So you can call this slice number 1, this is slice number 2, this is slice number 3. Okay, so when slice number 2 is depolarizing, right, this slice number 3 is still resting, and slice number 1 is in refractory. So even though sodium is diffusing in both directions, can the inside of slice number 1 get more positive? No, because it's automatically being pumped out during refractory. So in this way, the action potential only moves down the neuron, right? It can't go backwards because the previous slice is always in refractory. So when the action potential moves now to slice number 3, what happens to slice number 2? It's in refractory. So in this way, the action potential always moves forward, never backwards. So in your notes, I've given you this diagram, and I have seen this question many, many times on diplomas. And if it's going to be on diplomas, it's going to be on your quizzes and your tests, right? So what it's showing here is that slice of the neuron. And it's showing kind of what's happening in the different sections. So if we have our action potential, right? So our action potential we've represented in graph form kind of like this, right? We know the different sections: resting, depolarization, repolarization, hyperpolarization, back to resting. Hopefully at this point you guys know what's going on in each one of those parts of the graph. Okay? So you know that when a section of the neuron is depolarizing, that's when our sodium is rushing in. So the middle section here is depolarizing. It's in this part of the curve right here. The slice of the neuron right ahead of it is in resting. So this part right here is resting. And the previous part of this particular little section that we're looking at had just gone through depolarization, and it is repolarizing. It's in refractory. All right, so this section right here is at this part of the graph. And in this way, the action potential is going to continue to move forward. It cannot move backwards because the section of neuron that just depolarized immediately goes into refractory. Okay, and it can't generate another action potential for about 2 milliseconds. Now, when we talk about this, it seems like it's really slow, right? It seems like this is a really slow process, but we're talking thousandths of a second here, you guys, right? I can tap you on the shoulder really, really quickly, and you feel each individual tap. So the analogy that I like to think of is a fuse burning. Okay, if you think of a fuse burning like a neuron, right? When you light the fuse at one end, it only burns in one direction. Can the fuse burn backwards? No, right? It has to only go in one direction. And the fuse burning, that's the analogy for the traveling action potential. All right, so think of that bright light there in that little animation. That's the action potential. And it only travels down the neuron in one direction. Okay, it can't go backwards, just like a fuse cannot burn backwards. Okay, so the diagram above it kind of shows what's happening as far as the charge goes, right? Sodium rushes in, it diffuses forward. That causes threshold. More sodium rushes in, it diffuses forward, etc., etc.
This length of axon can be likened to a piece of fuse. The burning match under the axon represents an electrical event that generates an action potential. This event normally takes place at the axon hillock. Recall that to trigger an action potential, the electrical event must depolarize the axonal membrane beyond its threshold. That is, the membrane potential has to become positive enough to initiate the action potential, which is an all-or-nothing event. Once the membrane reaches that threshold, voltage-gated sodium channels open up, and sodium rushes in, as reflected in the upward deflection of the membrane potential. But how is the action potential propagated down the axon? Look at the area just ahead of where the action potential has just fired. It is becoming more depolarized. A millisecond later, the positive charge associated with the first action potential has spread to an adjacent region of the axon. This area reaches its threshold, and this segment of axon fires an action potential. The area of axon that fired an action potential a millisecond ago cannot become depolarized now. It is refractory because its sodium channels are inactivated. In this way, the action potential propagates in only one direction, like a fuse. Now, 2 milliseconds later, the positive charge spreads to the next adjacent region of the axon, and it reaches its threshold and fires an action potential. The region of axon that fired a millisecond ago cannot be depolarized. It is refractory. The area of the membrane that fired 2 milliseconds ago has now returned to its resting potential, but it is too far away to be influenced by the current action potential. In this way, the action potential migrates, like the fire on a fuse, toward the axon terminal, where it is delivered to another neuron. Now let's watch the whole process at high speed.
Okay, so just like a fuse burning, and again, they used fire as an example of a stimulus, but this could be sound, this could be touch, this could be light. Okay, something that's going to stimulate a sensory neuron is going to cause some sodium to rush in, and we get depolarization if we hit threshold. Now, that sodium that rushes in diffuses forward, it causes the next section to depolarize, etc., while the previous slice is repolarizing and it's in refractory. Okay, so burning of a fuse is a good analogy. Another one I used to use is dominoes falling, right? When you push one domino over, it hits another domino and it travels forward. But you can't, the dominoes falling can't go backwards because the domino's already fallen. So that's kind of like this propagation of a nerve impulse. So it can only move to slices that are polarized, okay, that are resting. Now, if you'll recall from our first lesson, I mentioned that some neurons are covered in what's known as a myelin sheath. Okay, now not all neurons are covered in myelin sheaths. Mostly neurons that are in the peripheral nervous system, and some areas of the brain and spinal cord have neurons that are covered in this myelin sheath. What is a myelin sheath? A myelin sheath is composed of special glial cells called Schwann cells. So each one of these things right here, this right here, this right here, okay, this right here, this right here, those are individual Schwann cells. Okay, each one of those is an individual Schwann cell. In this particular diagram, there are 4 of them. All 4 of these Schwann cells together constitute the myelin sheath. Okay, now what is it? Well, these Schwann cells happen to be very fatty. They're very, very fatty, and when you look at them under a microscope, they actually look white, like fat. And the reason why they're there is to insulate the axon. Remember, you guys, charged particles don't like nonpolar particles, right? Fat is nonpolar, oil is nonpolar, and sodium and potassium, they're charged particles. So what happens is sodium that's outside here, when it tries to get through the Schwann cell, it's prevented from doing so. Now, there are gaps between the Schwann cells of the myelin sheath. So you'll notice here, there's a gap between the 2 Schwann cells. That's known as the node of Ranvier. And in these particular sections where there's a gap between the Schwann cells, sodium can. It's not interrupted and it can flow into the axon. Now, why do we have this? Why have we evolved this? Because it really helps the neuron in that it saves energy, because wherever there is a Schwann cell, there's no diffusion from the outside to the inside, so you don't need to have sodium-potassium pumps working. Remember, I said sodium-potassium pumps are really energy intensive. They require ATP all the time. So if you don't have to work them, you're saving energy, okay? The other thing that's important here is that only small sections of the neuron have to depolarize, so that makes the action potential travel much quicker, all right? So some anatomy here, you guys. Again, this diagram, there's always confusion between what's the difference between a Schwann cell and a myelin sheath. A Schwann cell is the individual glial cell that wraps itself. You guys know what a tensor bandage is, right? A tensor bandage, you might put it if your arm is sore, your leg is sore, and you wrap it around your arm. Think of these Schwann cells like tensor bandages. They wrap themselves around the neuron, okay? And by doing that, they insulate the neuron. No charged particles can pass through that area. All right, so each individual thing here is a Schwann cell, and together they make up the myelin sheath. Now, as I mentioned, the gaps between the Schwann cells are called the nodes of Ranvier. And in these regions, the axon is exposed and ions can pass through. So ions cannot pass through the Schwann cells, but they can pass through the nodes of Ranvier. So action potentials only, okay, big circle here, only occur at the nodes of Ranvier, not where the Schwann cells are. So what we get is action potentials jump from node to node to node to node. And large sections of the neuron do not undergo depolarization and repolarization. So you're skipping big parts of the neuron, which means that these nerve impulses travel way faster. For example, okay, so just to put some numbers to show you how much faster this is, in a non-myelinated neuron, so a neuron that does not have this myelin sheath, nerve impulses travel at about 0.5 meters per second, which is still pretty fast. But in a myelinated neuron, that moves to 120 meters per second. All right, way faster. So what's the advantage of having myelinated neurons in my peripheral nervous system? Well, remember you guys, we talked about reflexes, right? If I put my hand down on a hot stove, do I want that action potential to travel from my hand to my brain or my spinal cord real fast? Absolutely. I want it to get there as fast as possible so I can take my hand off that hot stove real quick. Neurons in your peripheral nervous system are myelinated because you want those action potentials to travel very quickly, okay? Because you have to react to your environment and reflexes need to be fast. Neurons that are packed in your brain, packed in your spinal cord, where the distance isn't that far, they tend to be non-myelinated. We can pack more neurons in if we don't have that myelin sheath.
Saltatory conduction. Conduction through myelinated nerve fiber. Due to the myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made up of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier which are unmyelinated. As depolarization cannot occur at the cells making up the myelin sheath, the wave of depolarization can only occur at the nodes of Ranvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as saltatory conduction, and it serves as a means of increasing the rate of propagation of an action potential. Not only does saltatory conduction increase the speed of impulse transmission by causing the depolarization process to jump from one node to the next, it also conserves energy for the axon as depolarization only occurs at the nodes and not along the whole length of the nerve fiber as in unmyelinated fibers. This causes up to 100 times less movement of ions that would otherwise be necessary, therefore conserving the energy required to re-establish the sodium and potassium concentration differences across the membranes following a series of action potentials being propagated along the fiber.
We can actually see where the myelinated neurons are and where the non-myelinated neurons are. So remember you guys, I said that Schwann cells and myelin is very fatty. Neurons that are covered in this fatty, the Schwann cell, tend to look white. Okay, so if we look at the nervous system, anywhere where we see this really white color here, so in there, this thing's called the corpus callosum. If we look at the nerves that are coming out of the spinal cord, these are spinal nerves, okay, you can see that they all look really white. Those are myelinated neurons, okay, and we call that white matter. Non-myelinated neurons, they don't appear white. In fact, they appear more gray in color, and that's how they get their name. Okay, we call these regions of the central nervous system gray matter. All right, so white matter, it appears white, and it's because of that fatty myelin sheath. Okay, and gray matter is gray because it lacks that myelin sheath. So what I want you guys to understand in today's lesson is how the nerve impulse or the action potential travels from the dendrites through the cell body, through the axon to the axon terminals. It only travels in one direction, all right? And there's kind of two different ways that action potentials travel through different types of neurons. If it's non-myelinated, the action potential has to travel through the entire neuron. If it's myelinated, the action potentials only happen at the nodes of Ranvier, all right? So you have some homework in your note package. Please take a look at those questions on page 14. And that's it for this lesson.